1. From the diagram, we can see that 0.1 mm corresponds to 40 divisions on the eyepiece graticule. From division 50 to division 90. 2. Let's find what 1 division equals. 0.1 mm equals 40 divisions. 1 division equals 0.1 mm divided by 40 is equal to 0.0025 mm. 3. Convert 0.0025 mm to μm. 1 mm equals 1000 micrometers. 0.0025 mm is equal to 0.0025 times 1000 equals 2.5 micrometers per division. 4. The chloroplast is 4 divisions long. Length is equal to 4 by 2.5 micrometers. Length equals 10 micrometers. Length equals 1.0 times 10 superscript 1 μm. 5. Looking at the options. A. 1.0 times 10 superscript 1 μm. B. 4.0 times 10 superscript 2 μm. C. 2.5 times 10 dash superscript 1 μm. D. 2.5 times 10 dash superscript 2 μm. Therefore, the chloroplast is 1.0 times 10 superscript 1 μm long, option A. Let's convert each option to μm, micrometers. A. 1 nanometer to 5 times 10 superscript 2 nm. 1 nanometer equals 0.001 micrometers. 500 nanometers equals 0.5 micrometers. Range, 0.001 micrometers to 0.5 micrometers. B. 1 times 10 superscript 3 nm to 5 micrometers. 1000 nanometers equals 1 micrometer. Range, 1 micrometer to 5 micrometers. C. 1 times 10 superscript 1 μm to 5 times 10 superscript 2 μm. Range, 10 micrometers to 500 micrometers. D. 1 times 10 superscript 2 μm to 5 times 10 superscript 3 μm. Range, 100 micrometers to 5000 micrometers. Prokaryotic cells, like bacteria, are typically between 1 to 5 micrometers in diameter. Therefore, option B is the correct answer as this range, 1 to 5 micrometers, matches the typical size of prokaryotic cells. The correct answer is A, 1, 2, and 3. Let me explain why nucleic acids are found in all three labeled structures. 1. Cell membrane, 1. Contains nucleic acids in the form of membrane-associated DNA and RNA. Particularly important during processes like cell signaling and transport. Also involved in the passage of genetic material during certain cellular processes. 2. Rough endoplasmic reticulum, 2. Contains messenger RNA, mRNA, involved in protein synthesis. Has attached ribosomes which contain ribosomal RNA, rRNA. Important site for protein synthesis and processing. 3. Mitochondria, 3. Contains its own DNA, mitochondrial DNA or mtDNA. Has mitochondrial ribosomes containing rRNA. Contains various types of RNA needed for mitochondrial protein synthesis. Essential for maintaining mitochondrial function and inheritance. Therefore, since nucleic acids, DNA and/or RNA, can be found in all three structures, A, 1, 2, and 3, is the correct answer. This confirms the complex distribution of nucleic acids throughout different cellular components, each serving specific functions in cellular processes. 1. Intron, A. Introns are non-coding DNA sequences found in eukaryotic genes. Bacteria, being prokaryotes, do not have introns. Their genes are continuous without interruption. 2. Telomere, B. Telomeres are protective structures at the ends of linear chromosomes. Bacterial DNA is circular, not linear. Therefore, bacteria do not have telomeres. 3. Template strand, C. The template strand is one of the two DNA strands used during DNA replication and transcription. All cells, including bacteria, have template strands in their DNA. This is essential for DNA replication and gene expression. 4. Capsid, D. A capsid is the protein coat that surrounds viral genetic material. It is a viral structure, not a bacterial cell structure. While viruses can infect bacteria, capsids are not part of bacterial cell structure. Therefore, option C is the correct answer. Template strands are fundamental to DNA structure and function in all living cells, including bacteria. While the other structures are either eukaryotic features, introns, telomeres, or viral structures, capsid. 1. Structure of a water molecule H2O. Has 2 hydrogen atoms. Has 1 oxygen atom. Has 2 lone pairs of electrons on the oxygen. 2. Hydrogen bonding capabilities. Each H atom can form 1 hydrogen bond with the lone pair of another water molecule. 2 H atoms equals 2 hydrogen bonds as donor. Each lone pair on O can accept 1 hydrogen bond from another water molecule. 2 lone pairs equals 2 hydrogen bonds as acceptor. 3. Total maximum hydrogen bonds. As donor, 2 bonds, from H atoms. As acceptor, 2 bonds, from lone pairs. Total, 2 plus 2 is equal to 4 hydrogen bonds. Therefore, option D, 4 is the correct answer. Each water molecule can participate in a maximum of 4 hydrogen bonds. 2 through its hydrogen atoms, acting as H-bond donor. 2 through its oxygen's lone pairs, acting as H-bond acceptor. This tetrahedral arrangement of hydrogen bonds is what gives water many of its unique properties, including its high boiling point and surface tension. Let's analyze each amino acid. Glycine. NH3. Plus group, +1. COO group, -1. Net charge equals 0. Lysine. NH3. Plus group on alpha carbon, +1. Additional NH3. Plus group on side chain, +1. COO group, -1. Net charge equals +1. Alanine. NH3. Plus group, +1. COO group, -1. Net charge equals 0. Aspartate. NH3. Plus group, +1. COO group, -1. Additional COO group on side chain, -1. Net charge equals -1. 2. Looking for amino acids with overall charge. Lysine has +1 charge. Aspartate has -1 charge. Glycine has 0 charge. Alanine has 0 charge. Therefore, option C, aspartate and lysine, is the correct answer, as these are the only 2 amino acids that have an overall charge in aqueous solution. 1. For triglyceride 56:4, we know. Total carbon atoms equals 56. Total double bonds equals 4. It contains 3 fatty acids, 17:0, X, and 18:2. 2. Let's find fatty acid X. For carbon atoms. Total equals 56. Fatty acid 1 has 17 carbons. Fatty acid 3 has 18 carbons. The glycerol backbone has 3 carbons. Therefore, 56 minus 17 minus 18 minus 3 is equal to 18 carbons for X. 3. 4 double bonds. Total equals 4. Fatty acid 1 has 0 double bonds. Fatty acid 3 has 2 double bonds. Therefore, for -0 - 2 is equal to 2 double bonds for X. So fatty acid X must be 18:2. 18 carbon atoms. 2 double bonds. The presence of double bonds means it's unsaturated. Therefore, option D, unsaturated with 18 carbon atoms, is the correct answer. 1. Let's identify what each structure level means. Primary structure, the sequence of amino acids. Secondary structure, regular local folding patterns. Tertiary structure, overall 3D folding of a single polypeptide chain. Quaternary structure, interaction between multiple polypeptide chains. 2. From the description given. Regular pattern of amino acids, and every third amino acid is glycine, describes the primary structure. 3 polypeptide chains interacting together, describes the quaternary structure. 3. This combination matches option B, primary and quaternary. The regular pattern of amino acids is a characteristic of the primary structure, while the interaction of 3 separate polypeptide chains to form the complete collagen molecule is a characteristic of quaternary structure. Therefore, the answer is option B, primary and quaternary. Note, while collagen does have a unique secondary structure, triple helix, and tertiary structure, these are not specifically described in the given information. 1. First, let's identify the structure of a hemoglobin molecule. It has 2 alpha-globin chains. It has 2 beta-globin chains. It has 4 heme groups, 1 for each globin chain. 2. Let's calculate the total mass in Da. 2 times alpha-globin equals 2 times 15,126 is equal to 30,252 Da. 2 times beta-globin equals 2 times 15,868 is equal to 31,736 Da. 4 times heme group is equal to 4 times 617 equals 2,468 Da. 3. Total mass in Da. Total equals 30,252 plus 31,736 plus 2,468 is equal to 64,456 Da. 4. Convert to kDa. Given, 1000 Da equals 1 kDa. 64,456 Da equals 64,456 divided by 1000 is equal to 64.456 kDa is almost equal to 64.5 kDa. Therefore, the mass of a hemoglobin molecule is 64.5 kDa. The answer is option D. 1. For the production of an extracellular enzyme, we need to consider the correct sequence of cellular processes. First, transcription occurs in the nucleus where DNA is used to make mRNA. The mRNA then moves to ribosomes, which are attached to rough ER for proteins meant for export. The protein is synthesized and enters the rough endoplasmic reticulum. Finally, the protein moves to the Golgi body for modification and packaging for secretion. 2. Looking at the options. A. Nucleus ribosome rough Golgi body. This is the correct order because DNA is transcribed to mRNA in the nucleus. mRNA goes to ribosomes. Protein enters rough Protein is processed in Golgi body. B, C, and D all have incorrect sequences that don't match the biological process. Therefore, the answer is option A, nucleus ribosome rough endoplasmic reticulum. Golgi body, this represents the correct pathway for the synthesis and processing of an extracellular enzyme.